All right. So thanks everyone for coming. I'd like to welcome you all to the keynote talk of Day 2 of Darwin Week 2012. Before I get started, I have just a few really quick housekeeping notes. On your chairs, you'll see that there are speaker rating cards. You can fill those out during or after the talk or whenever. You can leave them on your chair if you want, or you can take them to the back table. There are also bookmarks. They should have a schedule for all of tomorrow's events. So hopefully we'll see some of you there. And finally, and maybe most importantly, there's also some free food back there. There's cookies and pops, so please take it because we don't want to have to deal with all of that after Darwin. So with that said, I'd like to introduce our speaker tonight. Dr. Clint Kelly is an assistant professor in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. He teaches courses in animal behavior and Darwinian medicine. His research interests include sexual selection, sperm competition, and the evolution of sexual dimorphism and eco-immunology. Please give a round of applause for Dr.
Thank you very much. I'd like to thank everybody for coming tonight. And I'd like to thank UNIFI for inviting me, and in particular Joe for organizing my visit. And it's a great honor for me to be speaking during Darwin Week, given that Darwin is one of my scientific heroes. So today I hope to present a series of case studies illustrating the power of sexual selection to create dramatic morphological diversity in sperm and in genitalia. And I hope you walk away from this talk with a better appreciation of not only the power of sexual selection, but also the wide array and diversity of morphologies we see in the natural world. And given that it's Valentine's Day, female partners may be looking askance at males by the end of this. So hopefully no serious mishaps. So, as with all tales of evolution, our story begins with Charles Darwin. Darwin was brilliant. There's no question about that. But he often became puzzled over a few things. And one of those things that really puzzled him was the presence of exaggerated traits, particularly in males. These colorful displays, large ornaments, big armaments. These traits puzzled him because they didn't increase the bearer's survival. In fact, they often decreased the bearer's survival. But with considerable thought and some experiment, he quickly realized that, hey, these traits may reduce survivorship of an individual, in most cases the male, but at the same time they increase reproductive success. And at the end of the day, it doesn't really matter how long you live as long as you put more kids into the next generation than your competitors. That's fitness. So, as he puzzled these things in his book, Descent of Man, he came up with this wonderful quote. "We are, however, here concerned only with that kind of selection which I have called sexual selection. This depends on the advantage which certain individuals have over other individuals of the same sex and species in exclusive relation to reproduction." This is essentially the same definition that biologists, practitioners of sexual selection theory use today. So, sexual selection is generally put into two categories. We have intersexual selection, and this commonly involves females choosing males. And this process typically ends up in something like this. The male peacock, that's redundant, it's a peacock, train being a very large, colorful, extravagant trait. And females choose males that have larger trains, more colorful, and often more symmetrical eye spots. So sexual selection has driven the evolution of this particular trait, and other traits like it, like the song of the cardinal, or even pheromones. So some moths, males produce good-smelling stuff that females find attractive. So we've got intersexual selection, or female mate choice. And we've also got male-male competition, or intrasexual selection. Intra meaning within the same sex. And typically, again, this is within or between males. And a classic example of that are the enlarged antlers of ungulates. So males use these traits, these structures, in fights with one another over access to females. And males with bigger weaponry tend to have better access to more mates. So we tend to think about armaments and ornaments and their use in sexual competition before copulation. So this is where we see the stuff happening. We see males display in front of females. We see males fight one another for access to females. But there is as much going on during copulation and after copulation as before copulation. And one of those sorts of competition is something we call sperm competition. It's a very simple definition, and I'll elaborate on this later on. It's simply the competition among sperm from different males to fertilize the ova of the same female. And so you might ask yourself, well, how do sperm compete? They don't have big trains, they don't have songs, they don't have large weapons, they're not fighting one another in a typical sense, like you'd see two red deer fighting one another. They fight through more, through different means, something we call displacement. And this form of competition, displacement, is common among animal taxa. And it essentially involves males pushing other males out. And here we see the inside of a female Drosophila fruit fly. Here we see the seminal receptacle in a female fruit fly. This is where the sperm end up, and this is where the females tend to pull out the sperm to fertilize their eggs. So with this remarkable technique, honed in Scott Picknick's lab at Syracuse University, they are able to genetically sort of modify males and have some males produce red sperm and some males produce green sperm. And this has allowed us to actually see sperm displacement and sperm competition in action. We used to use, or we still use, other means, more indirect methods of figuring out exactly how sperm are competing within the female. But this gives us a really direct method. And there's a bit of a glare here. So what we see is a female that was mated first to a male that produces green glowing sperm. And then a short while later, she was mated to a male that produces red glowing sperm. And if you watch this loop, you see that the red sperm are coming in up here, going around. They're coming up here, swirling around. And the green sperm are being pushed out. It's just sort of a simple matter of fluid dynamics. The new, bigger ejaculate is washing in, and just like a wave, pushes those other sperm out, and they exit and are not available for fertilization. So some green sperm will stick around, but it'll most likely be mostly the red male sperm that'll be available for fertilizations. This was all, this would be quite a shock to Darwin, I think. There is quite a debate amongst historians of biological sciences as to how much Darwin knew about what kind of selection was going on after or during mating. So some historians believe that Darwin, like many biologists at the time, were convinced that females are generally monogamous. All that action happens before copulation. Females are choosing, males are fighting. Female chooses a male, mates with that male, takes his sperm on board. She's done. She fertilizes her eggs with those sperm, lays the eggs, and that's that. So there wasn't even sperm competition happening because you need two ejaculates from two males to have sperm competition. Other historians have picked out some little bits in his writing that tend to suggest that he knew exactly what was going on. And really, Darwin was such a keen observer. It would be hard to believe that he was oblivious to something so obvious. But these historians suspect that he already came out with theory of evolution by natural selection. He's coming out with sexual selection, which is already pretty racy. Wow, males displaying for females, and females choosing males in this upstanding Victorian society. So these historians figure that coming out with sperm competition between ejaculates within female, that would just be piling on. And that would definitely upset Victorian sensibilities, even more so than his general theory of sexual selection. So it's believed that he probably kept his cards close to his chest and didn't reveal that this was going on. So we don't really know exactly what Darwin knew at the time about selection occurring during copulation and after copulation. Musical interlude. Is that coming from my bag?
Is that yours?
How embarrassing. It's not even my phone. Yeah, that would be my wife's phone in my bag. She was calling to find out how much the ticket was for speeding on the way here. Yeah. The I-35, they're waiting right there. So we don't know exactly what Darwin knew at the time, but we fast forward to around late 60s, fitting, some talk about sperm competition, sexual revolution. Along comes this brilliant biologist by the name of Jeff Parker, now Sir Jeff Parker, who was knighted a couple of years ago. And he published a paper entitled Sperm Competition and Its Evolutionary Consequences in the Insects. And this was the first time anybody sort of critically thought about what was happening within the female when two males were copulating with their competing ejaculates. And in this paper, he essentially started a very active and large research program that is incredibly vibrant to this day. And so essentially in this paper, he outlined various hypotheses and predictions as to what should happen with regards to how selection should shape male reproductive behavior and ejaculates and sperm in animals, given that females are mating with multiple males. And the groundwork for his hypotheses was that, counter to common knowledge or common thinking, sperm are costly to produce. They're not cheap. And whenever you have something that's costly to produce, you should spend that rather prudently. And so Parker suggested that males producing sperm should spend these ejaculates prudently based on various bits of information that influence their paternity success. These bits of information include female quality. They include the mated status of the female, whether she's a virgin or mated, and how many rivals are out there that could possibly be competing with this ejaculate. So fast forward to last year, a colleague and I meta-analytically reviewed hundreds of studies of of males tailoring their ejaculate in real time with regards to various socio-ecological circumstances. And we found strong support for Parker's hypotheses. Males transfer more sperm to higher quality or bigger, more fecund females. They transfer more sperm to virgin females. And they transfer more sperm in the presence of a rival. So essentially, males transfer sperm in a fashion that maximizes their probability for fertilization. So this is one aspect of how sexual selection tailors ejaculates. This is a facultative adjustment, let's call it. So this is a male tailoring his ejaculate in real time, right now. The other effect that sexual selection has on sperm is more long-term, more evolutionary, and more obligate. So sexual selection can cause these real-time changes, but it can also cause more permanent changes to the sperm in terms of morphology. So essentially every male produces, or every male in a species produces roughly the same type of sperm. And one of the constants, or one of the general patterns across animals is that sexual selection favors longer sperm, bigger sperm. And this makes sense when you think that sperm length dictates how fast a sperm can travel. So longer sperm are faster. And when you're in a race to get to the egg before other males' ejaculates or other sperm, you want to swim fast. So across animal taxa, we see these positive relationships between the intensity of sperm competition, and biologists generally use relative testes mass as sort of a surrogate or proxy for the intensity of sperm competition. And in this particular study on eutherian mammals, we see that the intensity of sexual selection is positively correlated with a number of sperm traits: head length, total sperm length, mid-piece length. That's where the ATP lies. That's the energy package in the sperm. So all these little bits and pieces of the sperm are positively correlated with the intensity of sexual selection. So more competition for eggs drives the evolution of longer sperm, and those longer sperm are faster and they get to the egg before competitors. And this pattern is repeated across animal taxa. Here's another study I just pulled out on shorebirds. So in shorebirds, species that encounter more intense sexual selection also have longer, faster sperm. And we even see this in African cichlids. John Fitzpatrick and his colleagues a couple of years ago published this really nice paper, and they went one step further, where they actually showed that longer sperm are indeed faster, and those sperm occur in species that have really intense sexual selection. So in this particular species, they have small, slow sperm. They're a monogamous species. So one male, one female, they raise a brood together and then move on. This species has really intense sexual selection. They have 4 male mating morphs. So they've got regular males, dominant males. They've got female mimics. They've got little sperm or sneaker sperm males that dart through the nest and deposit sperm. So there's competition coming from all sides. And so when a female is laying eggs, you need to have your sperm there now. So they have evolved really long, fast sperm. So across animals, we see this increase in sperm length with regards to intensity of sperm competition. This all culminates in the fruit fly, Drosophila bifurca. They've got the longest sperm of any animal on the planet. Any guesses as to how long their sperm might be? Throw out a number. Anyone? 6 centimeters long. That's equivalent to a 6-foot male producing a 120-foot long sperm. So you're standing on the goal line, it's at the 40-yard line. That's a long sperm. So males produce approximately 200 of these a day, and they still outnumber female eggs by a ratio of about 6 to 1. Compare that to the little fruit fly you find on your bananas. That's Drosophila melanogaster, and those males produce about 1,750 sperm a day. What's also remarkable is that only a very, 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 very tiny fraction of that sperm, the very tip of the sperm head, enters the egg. The rest is all sort of discarded. Those nutrients don't get reabsorbed by females. It adds nothing to male fertilization abilities. It really does nothing except propel the sperm up the female sperm storage tubules, and essentially, for a while anyway, blocks those. So whenever a male mates with a female, he may not be inseminating many sperm, but he's going to be fertilizing all the eggs that are available by the female. When you contrast that with Drosophila melanogaster, which have much smaller sperm, which are still about 11 times longer or 10 times longer than a male human sperm. So in those little fruit flies you find on your bananas, those males produce lots of sperm, but there's also multiple ejaculates from multiple males can fit into a female, as we saw in that little slide with the red and green sperm. And so you end up having to share paternity in a brood. So there's a trade-off. And this represents a really nice trade-off between the classic size and number relationship. So these guys produce enormous sperm, but they can only produce few of them. And for reference, here's an egg. This is a crazy shaped egg. Here is a single sperm in a ball, and I think that's the head right there. And this, just for perspective, this is that sperm compared to the female egg. So females produce 1 egg for every 6 sperm produced by the male. So sexual selection favors the evolution of longer sperm, but it also has pretty remarkable effects on the morphology of the sperm head as well. And in a study published a couple of years ago by Harry Moore and colleagues on the wood mouse, this was just sort of a serendipitous find by Harry Moore. Apparently his cat brought in a wood mouse. This wood mouse had enormous testicles, and so being a good biologist, he recognized, hey, there's probably pretty intense sperm competition going on here. So he dissected up the testes, had a look at the sperm, and recognized that it has this crazy morphology with this hook at the head. And so he dissected them out, looked at the sperm in a medium under the microscope, and he found something quite remarkable. These sperm would glom on and hook on to a brother's sperm. And sometimes they would hook on to the head of a brother's sperm, but most often they would grab the flagellum of another sperm and hang on. And this little hook here would sort of clamp shut, dunk, and hold on to that flagellum. And this would happen hundreds and hundreds of times. And pretty soon he found that the sperm were producing these long sperm trains, and they would start to propel their way across this microscope slide. And what's also remarkable, later on they found that whenever a sperm clamps onto the flagellum of another sperm, its acrosome becomes incapable of penetrating an egg. So essentially, by clamping onto a flagellum, it becomes an infertile sperm, and the only sperm that's able to fertilize the egg is the one at the head of the train. So this is kind of the ultimate in cooperation. But not really, because when you think that these are all haploid cells, so they're all related to one another at the same rate, so it doesn't really matter whether it's me that fertilizes the egg or one of my little brother's sperm. We're all putting roughly the same genes into that egg. So they extended this work, and they looked at sort of the function of these trains. So they thought, you know, across species, it's good if you are experiencing intense sexual selection to have really long sperm. Maybe this is, instead of producing single long sperm, this species gets around that by joining up everybody and having better propulsive forces by being in a long train. So they tested this idea, and they put sperm that form trains and single sperm into liquids of different viscosity, and measured how long it took them to travel a certain distance. And indeed, they found that sperm that form trains travel significantly faster in all viscosities than single sperm. So if you're a female wood mouse, you go out, you are in estrus, you mate with a couple males, those males' sperm are going to form trains and start sort of competing on different tracks up to the ovum. And the longer the train, the more propulsive force, the more likely that male is to fertilize the egg. And again, they extended this work and looked across rodent species. So we see these mice scurrying around our kitchens. You think, well, they don't do anything interesting except eat our bran and fiber. But in fact, they do some pretty cool stuff. So they looked across 37 species and examined the head of the sperm and acquired a couple different measures of the shape of that head. And they found, in both cases, the shape index and the angle of the hook, they found in both cases that there was a significant positive relationship between the shape of the sperm and the intensity of sperm competition. So they hypothesized then that by having a better hook or a steeper angle, those sperm are better able to grab the sperm and hold on, and thereby make longer trains. And that'd probably be the next step in the research, is to compare how long these trains are and how good they are at moving through the female's reproductive tract. But that's pretty strong evidence that sexual selection, sperm competition, is driving not only length, but sperm morphology, the morphology of the sperm. So up to now, we've talked about the ejaculate, we've talked about sperm. So hopefully I've demonstrated that sexual selection can have pretty strong effects on male sperm length, and it can also have pretty strong effects on male sperm morphology. Now I think we should turn our attention to the device that delivers the sperm, which is the penis, or phallus, or intermittent organ, or adiagus, whatever you choose to call it. Up until about mid-1985, most biologists considered the penis just a very simple functional organ. It delivers sperm. Some taxonomists, people that identify species of individuals, use them in their classifications. It's a really, penises tend to be unique to a species, and they're really good means of identifying an individual to a species. So up until about mid-80s, that's what people thought penises were. Something fun to play with too, I guess. Mid-1980s, about 1985, Bill Eberhard, which is an amazing biologist, naturalist who does most of his work out of the Smithsonian Tropical Research Institute in Panama. He published a book on animal genitalia and how sexual selection can drive the evolution of genitalia and their wild and dramatic morphological diversity. So he published his book, and then through the 90s, Goran Arnquist added to Eberhard's work, and together they came up with three main hypotheses to explain the evolution of genital elaboration or genital diversity. One of those is cryptic female mate choice, the other is sperm competition, and the other is sexual conflict. And I'll go through each of these in turn. So with regard to cryptic female choice, it's quite a simple definition. It's the capacity of females to control by various internal, hence cryptic, mechanisms the fertilization probabilities of their eggs by sperm from different males. So typical female mate choice, we see a female choose among males that are courting in front of her. But, again, stuff happens inside. And often we find that females that are better stimulated, tickled, pleased, etc., by particular males choose those males as mates and bias the paternity of their offspring to those males. Sort of the best ticklers get the offspring. This is a difficult hypothesis to test for a number of reasons. But one really nice bit of work was published recently by Clarissa House and Lee Simmons at UWA in Australia. And they asked the question, do male dung beetles that have particular penile morphologies, do they have greater paternity or fertilization success than other males? And over here you see the inflated phallus of a dung beetle, and this is sort of a clear tissue, and on top of it are these sclerites in black. And those same sclerites are shown in cartoon form over here. So using a number of mating experiments and molecular techniques to assign paternity to offspring, they found that males that had a particular penis morphology had greater mating success. In particular, males that had a shorter sclerite number 1, which is this bit here, and a larger sclerite number 2, which is this bit here, had significantly greater paternity success. So these particular traits, these particular structures, do something inside the female that makes her bias paternity towards males. And those females that they were using were mated by multiple males, and all those males had different sort of penis morphologies, and it was those that had the shorter sclerite 1 and the larger sclerite 2 had greater fertilization success. This hypothesis, the cryptic female mate choice hypothesis, was also supported by Dixon and Varel when they looked at various primate species and their penis morphologies. So we've got, on the left-hand side here, we've got some pretty spectacular penis morphologies. These are, there's lemurs in here, lorises, tarsiers, chimps, gorillas, humans, over here. You might want to see a doctor. So happy Valentine's Day, honey. I don't know if that guy's going to be very popular. So what Dixon and Varel hypothesized was that these structures don't do anything with regards to reproduction per se. What they do is they stimulate the female. And those males that better stimulate the female are awarded more paternity by that female. And so they predicted that you would find more spinosity, more tubercled penises in species in which females mate with many males. So if a female's mating with many males, you want to be the one to stimulate her best and then thereby get those offspring. Versus monogamous species. We don't really, these species, I shouldn't say we, don't want to pleasure the female as much as these guys. So this hypothesis that penis morphology was related to the mating system was supported for a while, but more data was collected and was found to be rather weak with regards to that particular hypothesis. Then along comes Paula Stockley in 2002 and readdresses this hypothesis by using a different response variable. She very cleverly noted that these species all differ in the period of time that females are receptive. So females have an estrus period, but they're not sexually receptive during that entire time. Some species have a estrus period of many days, but are only sexually receptive for maybe 8 hours. And so what it turns out, what she hypothesized was that males that had these penises with more accoutrements on them didn't evolve to pleasure the female. It was quite the contrary. They evolved to irritate the female. So essentially, Paula suggested that these traits evolved in order to abrade, irritate, aggravate the female's reproductive tract so she wouldn't mate again. Whereas these didn't have to. They're essentially, most of these species are monogamous. They mate with one individual. It's not really an issue. So she collected these data and found that indeed, there's a much stronger correlation between the female's receptive period and the morphology of his penis. So that falls into the category of sperm competition. As I said before, sperm not only compete, that's one way that males can compete with other males, but with regards to sperm competition, males have a number of behaviors that they, essentially at the end of the day, they just want the female not to mate again. They don't want any more sperm taken on board. So males, one form of sperm competition resistance is for males to guard females, not allow her to mate again. And that example that I just showed with the primate penises is one way of sort of guarding the female, just make her too sore to mate again. But there are other ways that males can engage in sperm competition. They can guard the female, they can compete one-on-one with the sperm of another male, or they can just simply remove the previous ejaculate. After reading Parker's 1970 paper, Jonathan Wagg who studies odonates, or dragonflies and damselflies, came up with the idea, after looking at his study species under a microscope, he came up with the idea that male damselflies compete with one another in the sperm competition arena by removing the ejaculate of the previous male. So damselflies and dragonflies have these devices on their penises, these recurved spines. There's a number of different types of structures on the penis. And essentially, a male inserts his penis and pulls out up to 90 to 100% of his rival sperm. And so here we see this hook here with male rival sperm there, and here the sperm is hanging off the penis here. So it's a pretty effective way of outcompeting your rivals for access to the female's eggs. And it's just not limited to spines. In this particular species, males have a brush. It's almost like a shoe brush at the tip of their penis. And they insert it. It sort of acts like as a mop. And it sucks up the previous male's ejaculate. And as they are pulling out, they insert their own sperm. And this could go on and on. So you just hope that you're the last male to mate. And one of the beauties of one of the beautiful things about evolution is once natural selection or sexual selection hits on something that works, you tend to see this repeat across taxa in sort of convergent ways. And this Argentine lake duck also has utilized the old brush at the end of the penis trick. And it's got this little brush down here that is hypothesized to remove sperm from previous males. Now, the Argentine duck has the largest penis among birds. Most birds don't have a penis, they just have a cloaca, and they mate with a female by performing something called the cloacal kiss. So they just sort of touch behinds, and the male, inserts sperm. These guys actually have a corkscrew penis, and this penis is 42.5 centimeters long. It's pretty remarkable. So the authors of this paper, we don't really know how this device evolved. Some are happy about it, though, I bet. So one hypothesis is that it evolved to be better at removing the sperm from previous males, the sperm competition hypothesis. And that's supported by this little brush down here. But what's also interesting in this species is there is a fair amount of forced copulation. So a male will land on a female and try and mate her against her will, and she resists. She kicks him and flaps her wings, and she takes off. And so one hypothesis is that this long corkscrew penis has evolved as a result of sexual conflict between the sexes. So the female's reproductive tract is corkscrew-shaped as well, and it's very long. And so one hypothesis is that in the ancestral condition, females had a short reproductive tract. Males, through male persistence, an arms race built up, and females evolved a longer corkscrew, males evolved a longer corkscrew penis. She evolved a longer corkscrew reproductive tract, male evolved a longer corkscrew penis. And so the penis length evolved as a result of this conflict, and perhaps sperm competition is responsible for this little brushed tip. So historically, we've always thought of male-female interactions as a collaborative collegial affair, where the intentions of both males and females were the same because they have the similar goals. But over the last few years, evolutionary biologists have realized that that's not always the case. Male fitness across animals is typically dependent upon mating frequency, or how many mates they get. Whereas female fitness often depends upon the quality of mates they get. So they've got divergent attitudes towards how many times they should be copulating. Males typically want to copulate a lot. Females want to copulate less often. So there's this little tug of war. And you end up with the evolution of this little feedback loop where males evolve traits that give them better persistence,
better able to inject sperm against the female's will into the female, and the females evolve better traits at resisting males, and around we go. So if we think about that duck from Argentina, female has a short reproductive tract, male evolves a longer penis, and around we go. Come back in a million years, maybe that penis is a meter long. I don't know how he'll take off from a lake, though, but quite the air drag. So speaking of sexual conflict, one of the best, quickly emerging classic cases is this little bug called the bean weevil, Callosobruchus maculatus. It's a very common model organism in behavioral ecology and evolutionary biology studies. It's rather nondescript, but when biologists had a look at the male's penis under a scanning electron microscope, they found this marvelous structure. And so the structure has like these little support braces, little spines here, and it culminates in this, what looks like a sea anemone at the top. And those aren't soft. Those are hard, rigid, spiky prongs sticking out the end. And so the obvious question is, why would they have those? It doesn't seem to act in any way to mop up a previous male's ejaculate. But what it does do is damage the female's reproductive tract. And this, again, is a scanning electron micrograph of the epithelial tissue of the female's reproductive tract. And these little arrows are pointing to these spikes. So we think of the penis coming through the screen. Not only is the penis, these little spikes pressing against that tissue, but they're actually poking through in various areas. So a male is not only mating with the female, he's causing her some pretty significant harm as well. And when you start looking at the literature across animal taxa, it's surprising that this happens more common than you would think. You would think that a male would want to have his mate be well and happy and be ready and available to lay as many eggs as possible. So this is sort of seems counterintuitive. Why would you damage the individual that's carrying your sperm and potentially going to sire your many offspring that will increase your fitness? Well, biologists have come up with two hypotheses to try and explain this seemingly odd phenomenon in the natural world. One is reduced female remating with rivals. We've already hit on this. Essentially, a male will scrape, scour, irritate the female's reproductive tract, so she's too sore to mate with rivals. And so the next time she lays eggs or has babies, she will use the sperm from that last male. The second hypothesis is that it causes females to lay eggs sooner. So a female incurs some damage, has this little feedback loop that says, uh-oh, we're in trouble, we may not live very long, we've got to oviposit or lay eggs, have babies right now. And so she does, and she uses the sperm from that last male. These hypotheses have been supported in a number of different animals, but when they were tested in this bean weevil, they found no support whatsoever. So on the y-axis here, we have females that had suffered less damage and females that suffered more damage. And on the x-axis, the number of females that remated after 12 hours. And there's no difference, there's no statistical difference between females with more genital damage than those with less genital damage. So males aren't harming their mates so they don't remate. Females will happily go and mate again. The second test, looking at fecundity, females with more genital damage have the same number of babies as those with less genital damage. So males aren't damaging their mates in order to stimulate them to lay eggs. So as of today, we really have no clear idea as to why males have evolved this particular trait and why it damages females. Perhaps we're in the middle of some sort of arms race where females are evolving better traits to resist this pokey, sticky, outy thing. So my final example of the miraculous evolution of penises across the animal kingdom is the Trinidadian guppy. This is work that I did for my master's degree a few years ago. The Trinidadian guppy is it's a remarkable animal. We see guppies in pet stores, but those are wildly exaggerated versions of what we find in the wild. Guppies are a really well-established model system for investigations of evolutionary biology. And they're really quite a phenomenal little animal. So we see up here at the top is a male, and we see down here a female. Males are colorful. They have bright orange patches, and females are drab. Males, unlike most fishes, have a penis. Live-bearing fish have penises. So those are the common pet store fish like mollies and swordtails have penises. And it's quite rare among fish. And that's called a gonopodium. So this is not life-size. So here we see the gonopodium, and it has a hook and a hood down here. So a male will swing this structure out and touch the female's gonopore, right here, little female vagina, right here, and insert a little sperm packet. And it gets sucked up into the female, and she uses those sperm to fertilize her eggs. And they're a live-bearing fish, so they give birth to live babies. This female is very well gravid down here, and she will give birth to many babies. And they're small fish. And actually, the world's record, I think, is 258 babies from a female guppy at the Shed Aquarium in Chicago. Weird bit of trivia. So why are guppies remarkable? Well, on the island of Trinidad, the streams can be divided into two basic categories. Those with low predation and those with high predation. High predation streams have these voracious pike cichlids that are really attracted to guppies, in particular that orange coloration of the male. Low predation sites have very few predators. So males or females aren't really under tremendous threat. Now, this concerns both males and females because males get matings from females by courting them. And males show off their bright orange fish plumage to the female by doing something called this sigmoidal display. And they vibrate in the water column in front of the female. And so you can imagine a bright orange thing flashing in front of you. If you're a predator, that's a lovely sign that dinner is ready. So in high predation streams, males tend to be a bit more drab. They don't have as much orange as low predation streams. You go to a low predation stream, and the males are really brightly colored. They're always displaying, and females are choosing among these courting males. And in those particular places, females have tremendous control over who she mates with. Now, the other remarkable bit of behavior is that males sneak copulate. They can circumvent female mate choice. And they can swim by the female, stick out the gonopodium, inseminate her, and keep on swimming without breaking stride. And that tends to happen more in the high predation streams, because everybody is on guard for these predators, and it's really dangerous for both the male and the female to be engaged in some courtship in the water column in front of these predators. So everybody's a bit coy, and males then tend to swim by these females, insert the sperm, and continue on swimming. So I hypothesized that if males are sneak copulating more in high predation streams, they should evolve structures that allow them to better sneak copulate. And clearly, being able to, or having a longer penis would allow you to get closer to the female and inseminate her more readily than having to get really close to her. So it improves sort of the drive-by or swim-by insemination process. And so I went to 5 low predation streams in Trinidad, collected males, and 5 high predation streams, collected males, and measured their penis length. Explain that to your mom when you're home for holidays. What do you do? What does your son do? That's even more embarrassing. So what does your son do? He measures fish penises. Great. Right education, Wilson. And so at the end of my research, I supported my hypothesis. Males from high predation streams have significantly longer penises than males from low predation streams. So in effect, males from high predation streams are better able to sneak copulate females and circumvent female mate choice and thus reduce their risk of predation. So I hope I've convinced, or at least intrigued you, that sexual selection is a powerful evolutionary force that can drive the astounding array and diversity of morphological traits in sperm and in penises. Now, that Darwin did not intuit or expound on sexual selection's power to drive the evolution of sperm or genital morphology, actually today works to our advantage, because those of us that study these traits through the lens of sexual selection have a nice acid test for his theory of sexual selection. And day after day, week after week, year after year, we continue to robustly support Darwin's hypotheses of both natural selection and sexual selection. Thank you.
We have enough time left for a few questions. Before we do that, though, real quickly, I wanted to mention that after the talk, a group of people will be going to Beck's on the Hill to hang out. If you want to talk about this more or anything else that's going on, please feel free to join us. So do we have any questions?
You mentioned this recurring theme of females of species not wanting to mate and males wanting to mate. I guess I guess I'm having a hard time understanding, like, what's the evolutionary advantage, or is there one, of females of species not wanting to mate? Like, you would think that females who are more willing to mate would have more offspring, and that would.
Good question. So why? Let me rephrase and see if I'm right. Why would females resist mating if mating is a good thing to do? Is that kind of what you're?
Well, yeah. I mean, it seems like mating would be a good thing to do to pass on your genes to the next generation.
Right. The primary reason for this female resistance is sex is really costly. So sex involves injury, damage, increased predation risk, like we saw in the guppy. It also dramatically increases disease and pathogen transfer. It could also, in some circumstances, end up with females taking sperm on board from males that are less appealing or don't have good genes. So females are looking for quality, and they want to minimize the risks that they are subjected to. They're probably going to face risks whenever they mate, so they are really resistant to having those risks. Because in most species, the added factor is that in a lot of species, female fitness is tied to her longevity. So females can only crank out eggs at a certain rate, and the longer they live, the more eggs they lay per day, whereas a male can produce thousands of sperm in one day, mate, essentially, it could mate for 8 hours. A bunch of different females and be done. I'm finished. Whereas females are like, I gotta stick around for 30 days, thanks. Any other questions? Penises, come on. Yes. Size does matter.
Is there a limit to the advantage of the sperm length? Like, is there a point where it gets so long that it...
Don't know. That's a really good question. Where will it end?
Right.
So yeah, a fruit fly female can only have a coiled reproductive tract that's so long, and there's going to be some sort of limit to that. It could end up with one giant sperm sort of hanging out her back end at some point. Don't know. But that raises a philosophical point that some biologists have toyed with sort of in jest, but I think it has some serious implications. We define females and males as those that produce the bigger gamete, female, and those that produce the smaller gamete, the male. So it's kind of an interesting game to play and think, what happens in a million years when Drosophila males evolve a gamete that's bigger than the egg? Do we now call him or her because his gamete is now bigger than the female egg, if the female egg stays the same size? So it's kind of interesting to think about our definitions and categorizations of various biological phenomena. Yeah?
Yeah, you mentioned 3 reasons why males would increase sperm and number of sperm, and one of them was a virgin.
Yep.
How in the animal world does a male know if that female's a virgin? pheromones or?
At home Friday night watching X-Files. My wife's an X-Files fan, so I shouldn't. No, there are a number of ways that males can tell. So, for example, in the guppy, a male can look at a female, and if she has a flat belly, she may not have any offspring in her, so that may be a cue that she's a virgin. Higher probability that she's a virgin. In a lot of insects, a male can actually smell, take a sniff of a female, and determine whether a male has been on that female before. Let me see if I can call this up. Just give me a second. I've got a picture here that might, it's always worth 1,000 words, isn't it? And you might have a new appreciation for bedbugs after this. So the male bedbug has this penis here, and what biologists have recently found is that it has these little things down at the tip that they call pegs, and they were curious as to what those things were. And it turns out that when the male inserts his penis into the female's, what they call the spermolage, he can use these to detect whether the female's been mated before. So essentially he's tasting sperm. And they did this, they tested this experimentally by taking 3 groups of males, and in one group they had a control, and they just took some fluid from a female, painted it on the penis, and then stood back and asked how many sperm will this male produce. They took another group of males where they painted the penis from an ejaculate removed from the male, so another male. They took that other male's sperm, painted it on here, and asked how many sperm will that male ejaculate, and produced that many. And they took another group of males where they painted the male penis with the ejaculate of some other random male, and found that males produced that many sperm. So males produce significantly, or ejaculate significantly fewer sperm when they can detect that the female has mated before. So essentially they stick their penis in, have a taste, and then adjust how many sperm that they produce. Pretty remarkable, isn't it? And what's also remarkable is, let me back up here for a second. Is the reproductive system of the bedbug. So again, here we see that male penis, and it's got its little pegs at the end here. Here's the female, and here's her. Here's where the male inserts his penis. Bedbugs, female bedbugs, have a genital system in which they use to lay eggs, and they have something called a paragenital system, a place where they accept sperm, and this is called the spermolage. And essentially, it's this groove in her belly that the male takes his penis, slides along the belly, it falls in there, and he pierces her cuticle. This is known as traumatic insemination. So happy St. Valentine's Day to that. So the male inserts his penis into this little chamber under the female's belly, and the sperm then enter the female's bloodstream and make its way to her ovaries and fertilize the eggs. And it looks like this evolved, this apparatus here evolved after the male started performing his traumatic insemination. So in some ancestral bedbug, males were taking their penis and stabbing it wherever into the female, and those sperm would migrate to the ovaries. And in experimental tests, it's been shown that when you do that to a female, stab her in different places, it greatly reduces her lifespan. So the female said, and again, this is a nice example of these counter-adaptations. The female said, okay, you're going to stab me with your penis? I'm going to direct that penis to a particular spot, and have you inseminate in a little chamber in which I have very, very strong immune reactions. So females load up on red blood cells down in here, or hemocytes down in here, and a male inserts his penis, and if there's any bacteria or anything, they get gobbled up quite rapidly. And it also appears that females may be able to gobble up sperm from males that they're not that pleased with. So it's a pretty amazing little apparatus.
Okay, well, on that note, we'd like to thank Clint Kelly one more time for being here